Welcome to our poster presentation. The Vitro Micronucleus Assay detects DNA fragments or an extra chromosome in cells after cell division, indicating damages to the chromosomes. The US EPA has produced micronucleus datasets using a high-throughput approach. Very briefly, Chinese hamster ovary cells were treated with 292 chemicals on 96 well plates. For each chemical, tests were conducted with 20 concentrations, ranging from 0 to 200 micromolars. For every dose per chemical, data is recorded as percentages of micronuclei or hypodiploid counts. In this project, we propose a decision tree-based informatics approach to evaluate each chemical's micronucleus and hypodiploidy frequencies and classify them as positive, negative, and inconclusive. An in vitro to in vivo extrapolation analysis follows to assess the chemical's potential risk. In our approach, to be called positive for micronucleus, a chemical needs to satisfy the following conditions. First, micronucleus responses of 2 consecutive concentrations have to be greater than the 95th percentile of batch-specific solvent controls. Second, the observed increased micronucleus frequency needs to occur at non-cytotoxic or adequate cytotoxicity levels. Third, having a dose-response trend with a non-parametric test. Fourth, having a benchmark dose value lower than the highest concentration tested. An inconclusive decision is made when the first 2 conditions are false. The rest are considered negative. Applying this approach, we identified 157 unique chemicals with positive micronucleus response, 25 negatives, and 110 inconclusive. The same approach was applied to the hypodiploidy data and identified 209 positives, 27 negatives, and 56 inconclusives. There seems to be too many positives for hypodiploidy using this approach. Therefore, we attempted the approach proposed by Bryce et al. To assess hypodiploidy frequencies. In this approach, a positive call is based on at least 3-fold changes for micronucleus and at least 10-fold changes for hypodiploidy. We manually examined the 15 positive chemicals and verified that 6 were true antigens and 2 were potential antigens. The Bryce approach seems to be better, since many of the positives are established antigens. We then performed IVIV analysis to convert the BMDs of the positive clastogenic chemicals to oral administered equivalent doses and compare them to ExpoCast exposure estimates and ToxVal-DB traditional GenTox PODs from the US EPA ComTox dashboard. As you can see, the AEDs in yellow dots are more conservative than the traditional GenTox endpoints in blue and cancer PODs in black. We can then prioritize chemicals for further evaluations by computing the bioactivity exposure ratio, or BER. A lower BER is considered as having greater potential concern for risk assessment. In conclusion, we proposed a decision tree-based workflow to classify high-throughput micronucleus datasets, followed by IVIV analysis to assess their potential risks. Our proposed approach seems to work well with clastogens, while the approach proposed by Bryce et al. Seems to be better at classifying antigens. We believe our proposed informatics approach provides a useful analytical workflow to risk assessment applications. Thank you very much.